Okay, now we will talk about the embryology of uh, reproductive medicine. So, in assist fertilization, there is a very high technological level. With a microscope, we can pick up a single cell from an embryo and can perform the genetic analysis of the embryo. The possibility to take the sperm without ejaculation, um, so directly from the testes, is a very common procedure. Taking semen samples with high levels of DNA fragmentation, we can select the sperm without the DNA fragmentation with an antibody specifically directed against annexin 5, and it is a personalized medicine. Fertilization is a complicated event that occurs in the fallopian tube and implies different steps involving the identification and fusion of two gametes, which lead to the development of the embryo. Fertilization implies the maturation of the gametes, oocytes, and spermatozoa. Maturation: during the second week of gestation, primordial germ cells develop in the epiblast. During the following week, the third, they migrate towards the yolk sac. During the fourth week, these cells home to the developing gonads, where they arrive at the end of the fourth week. During migration and after arriving to the gonad, germ cells increase in number through a series of mitotic events. Once primordial germ cells arrive to a gonad of a genetically female subject, they differentiate into oogonia. These cells undergo a series of mitotic divisions, and at the end of the third month, they are surrounded by a rim of epithelial cells. At 4 weeks, oogonia are clustered in small groups in the cortical layer of the ovary. Most of them undergo meiosis, and some begin to halt in the prophylaxis of the first meiotic division, so primary oocytes. This at 4 weeks. 4 weeks. And at 7 weeks, most of the oogonia have transformed into primary oocyte, prophase of the first meiotic division. At this point, cellular death starts, and several oogonia and primary oocytes undergo atresia. The first 7 weeks, most oogonia have transformed into primary oocyte. At birth, oogonia are absent, dead or evolved to primary oocytes. From the gonad and primary oocytes arrested at the prophase of the first meiotic division are surrounded by a single layer of follicular cells. This is the primordial follicle, which is the primary oocytes plus a layer of follicular cells. All the oocytes have started the prophase of the first meiotic division and are blocked at the diplotent stage thanks to the production of oocyte maturation inhibitor produced by follicular cells. Anyway, there is some research that some researchers that state that we can have oocytes oogonia also in late ages, in the late um, in older women, adult women, ovarian germ cells in the ovaries of adult women. This, from a reproductive point of view, would mean that also women in menopause can have oogonia, but it's controversial. So, until the beginning of puberty, primary oocytes are maintained at primordial follicle stage. Then, every month, 15-20 follicles are selected from this group to undergo maturation throughout 3 stages. Primary oocytes begin increasing volume, and follicular cells become cubic and create a stratified epithelium of granulosa cells, primary follicle, Granulosa cells secrete a layer of glycoproteins on the surface of the oocyte, which constitute the zona pellucida. With the advancement of the development, granulosa cells form wide spaces full of fluid. When the secondary follicle is mature, the LH peak induces the completion of the first meiotic division, with the formation of the secondary oocyte and the first polar body. The cell then undergoes the second meiotic division but is blocked in metaphase few hours before ovulation. At this time, the pre-ovulatory follicle or graft follicle undergoes ovulation, releasing the mature oocyte that is captured by the fimbriae of the fallopian tubes. The second meiotic division will be completed only after fertilization, with the formation of the second polar body and the fertilized oocyte. So, just to repeat, you have three stages of the maturation of the oocytes. We have primary oocytes increase in volume, and follicular cells become cubic and create stratified epithelium of granulosa cell, primary follicle. Then, second stage, granulosa cells secrete a layer of glycoproteins on the surface of the oocytes, which constitute the zona pellucida. And third stage, with the advancement of the development, granulosa cells form wide spaces full of fluid. Then LH peak, formation of the secondary oocyte and first polar body. First meiotic division, formation of secondary oocyte and first polar body. Okay, the number of oogonia varies largely from the beginning of development to puberty. At 7 months we have around 7 million oogonia. At birth, primary oocytes range from 700,000 to 2 million. At puberty, we have around 40,000 secondary oocytes. In general, a woman has 500 ovulations, at least at present, while in the past women had less ovulations due to a lifetime due to more pregnancies and longer time of breastfeeding. The fact that nowadays women ovulate and menstruate more is linked to an increased prevalence of some diseases, such as endometriosis, present in 10% of women in these days, while our grandmothers were not affected at all. Nowadays, there are some studies that performed the gene expression profile of the granulosa cells and found that their profile correlates with the probability of the egg to be fertile, underlying their importance for the oocyte development. Fertilization events. First, we have maturation of the gametes. Then, simultaneous presence at the site of fertilization, the oocyte and the sperm have to meet in the fallopian tube. Sperm is ejaculated in the vaginal posterior fornix and it moves towards the upper genital tract. An ejaculate contains about 200-500 million spermatozoa. Vagina, cervix, uterus, and fallopian tubes are the regions where sperm can move and survive. These regions may also represent barriers to the sperm's survival. The vagina does not constitute a favorable environment for the sperm because of the acidic pH. After 2 to 6 weeks into the vagina, sperm shows unaltered motility and are blocked. Therefore, the movement to the uterus should be very rapid. Cervix and the cervical mucus, which is very elastic during ovulation, have many functions for the sperm transport. They favor the penetration of sperm during the ovulatory phase and block it in other phases thanks to mucus viscosity. Protection from the unfavorable vaginal environment favoring the sperm capacitation. Under estrogen action, the cervical os becomes larger, reaching 3 mm during ovulation, while viscosity and cellularity decrease. Sperm transport in the uterus is influenced by several factors, including sperm motility, but also by uterine contractions. It should be considered that sperm active movements is not critical, as also inert substances may reach the tubes. An experiment was performed by placing inert beads in a monkey vagina, and they found the beads in the peritoneum. If the oocyte is picked up by the fimbriae of the fallopian tube, it will meet the sperm there. After 10 to 20 hours after sperm entry, many leukocytes arrive in the uterine cavity, where they eliminate the remaining sperm. Third step is gametes identification. It involves different processes. First is capacitation, process by which a spermatozoon acquires the capacity of penetrating the different layers surrounding the oocytes. It occurs in females' genitals and it takes about 5-6 hours. It requires removal of surface glycoprotein and seminal plasma proteins from the spermatozoon plasma membrane. Then plasma membrane rearrangement with an increase in fluidity and permeability in order to favor the oocyte spermatozoon interaction, and then spermatozoon hyperactivation. The sperm requires a very active movement. After capacitation, we have spermatozoon oocyte interaction and binding through zona pellucida, through zona protein ZP3, then acrosome reaction with the release of acrosomal proteolytic enzymes, then membrane fusion, and finally male nucleus entry. Fourth step is gametes fusion, and fifth step is spermatozoon oocyte penetration. When the sperm enters the oocyte, there is the formation of pronuclei. Then there is male-female pronuclei contact, independent DNA replication in each pronucleus, pronuclear membrane break, chromosome condensation, spindle apparatus formation, which allows the rearrangement of the chromosomes and division completion. Some years ago, in the assisted reproduction lab, there was the idea not to touch the spindle when the sperm was injected into the oocytes. This is no more done since it was a long procedure and now the spindle is not controlled anymore. Once the oocyte is fertilized, we have the cleavage with the division of the oocyte into 2, 4, and 8 cells. These cells then compact to form a morula, and then after 5 days of development we have a blastocyst. So we have cleavage at the second, third day of development. There is a rapid subsequent mitosis with an increasing number of cells, the blastomers, which are symmetric, weakly bound, and non-communicating cells. Then compaction at the fourth day, the point of 1236 blastomeres, the embryo reaches the morula stage and compaction begins. We have increasing cell-cell contacts among blastomeres. They become more close, but they stay separated. We have active compaction. Blastomeres are more difficult to distinguish as single cells and are more adherent, and morula fully compact. It appears as a single unit with multiple nuclei. Some blastomeres or fragments could be excluded. Embryo can eliminate all those cells that do not pass the quality control, and this is very easily seen at the microscope with cells that flow out of the zona pellucida. And finally we have blastulation at the sixth day. From 32 to 36 blastomeres, a cavity filled with liquid starts to grow inside the embryo, and this will lead to the development of the blastocyst. Blastocyst is essentially made of 3 components. We have an inner cell mass, embryonic knob, trophectoderm, outer layer of flattened blastomeres, and a blastocele, the cavity filled with liquid. At the blastocyst stadium, the embryo undergoes hatching and then starts implantation. The oocyte is picked up as explained in the previous lesson and then is sent to the lab, where it's studied under the microscope. the oocyte is easily visualized and granulosa cells around it. On the same day, the male provides the semen sample, which should be evaluated. Okay, we should be evaluated. We can have a semen sample with normozospermia, that is normal semen, oligozospermia, which are spermatozoa concentration less than 20 times 10 to the 6th per milliliter, so less than 20 million per milliliter, astenozospermia, so that the motility is altered in at least 50% of the spermatozoa, teratozospermia, so that less than 30% of spermatozoa has normal morphology, oligoastenoteratozospermia, combination of the three defects, altered motility, concentration, and morphology. Azoospermia, absence of sperm in the ejaculate, aspermia, absence of the ejaculate. The semen sample must be capacitated with the capacity test. The semen sample is placed on a density gradient composed of molecules of high molecular weight. Based on the density, motile and alive sperm tend to go down while leukocytes and seminal plasma remain up high. Then the upper part is eliminated and the lower part is taken, where there are motile spermatozoa. After all of this, we have to choose between FIVET, which is the conventional in vitro fertilization, with a drop of sperm near the oocyte, leaving them for fertilization. This is performed in case of tube peritoneal factor, that is acquired or congenital tubaric disease, like tubaric occlusion because of a previous ectopic pregnancy, pelvic inflammation, or previous pelvic surgery. Also, it's performed in case of third or fourth grade endometriosis. Also in case of endometriosis if surgery didn't give any result or were considered not appropriate. Also in case of idiopathic infertility in the previous treatment. So insemination cycles. Idiopathic infertility if the previous treatment, excluding insemination cycles, didn't give results or it was considered not appropriate. Or therapeutic failure with first level techniques. Okay. ICSI is another technique that we can choose, where we inject the sperm inside the oocyte. This is performed with two pipettes, one to keep the oocyte still and the other to inject the sperm. This is preferred in case of severe grade male infertility, oligoastenoteratozospermia, obstructive and secretory oozospermia, so testicular or epididymal spermatozoa, or absent or reduced fertilization in previous in vitro fertilization cycle, or fertilization of frozen thawed oocytes, or poor quantity of available oocytes, such as a woman of 40 years old. Okay, we have an early blastocyst, that is a blastocele bigger than half of the volume of the whole blastocyst without increase in dimension compared to previous stadia. This is the embryo classification. We can have an intermediate blastocyst when the blastocele is bigger than the volume, than half of the volume, with a slight expansion and mid-thinning of the zona pellucida. We can have a full blastocyst when the blastocele is bigger than half of the volume with a marked increase in dimension and a very thin zona pellucida. We can have an hatching blastocyst, so no manipulations. We can have a fully hatched blastocyst, no manipulations. We can have hatching or hatched blastocyst after manipulations, so assisted hatching or biopsy. On the fifth day, we have a blastocyst, which is evaluated according to expansion, inner cell mass, and trophectoderm, which must have flat cells, and this last one is the best predictor of implantation. So, on the fifth day, we have finally the blastocyst. It's evaluated according to three parameters: expansion, inner cell mass, which should be as compact as possible, and trophectoderm, which must have flat cells. And this is the best predictor of implantation. When do we perform embryo transfer? Well, at day 3, cleavage state. It's preferred when there is poor oocyte retrieval, since the blastulation rate is approximately only 55%, a poor embryo quality, or an advanced maternal age. So, when there is poor oocyte retrieval, a poor embryo quality, or an advanced maternal age. At day 5, that is the blastocyst stage, it's chosen when there is an excellent embryo quality at the third day, better implantation window, and in case of a single embryo transfer. Nowadays, we also perform the pre-implantation genetic analysis with CGHRA. There are two possibilities. We can pick up a single blastomere from an intact embryo, but there is a high risk of damage. Or we can pick up cells of the trophectoderm, avoiding to damage the inner mass, which will form the fetus. A lot of embryos, oocytes and blastocysts are frozen. So when we stimulate women with high levels of estrogen, the endometrium is completely different from a physiological one. It's not very good in receiving the blastocyst. But if we freeze the blastocyst and leave the woman having a normal cycle and we implant later, the implantation rate is very high. Thanks to this method, 75% of good prognosis women achieve pregnancy after the blastocyst freezing.